In the name of GIGA, the Biomedical Research Center of the University of Liège and its tech transfer office Interface, I'm very pleased today to welcome you for what is already our fifth webinar of our series, GIGA Platforms, Key Actors for Your R&D. You probably may know GIGA is an interdisciplinary biomedical research center, but it's also a whole ecosystem between academic research, companies, and the University Hospital of Liège. That's to say that the GIGA platform gathers a whole range of complementary expertises and scientific equipment that are available to your academic lab or your company lab for a very customized service and collaboration. And by the way, if you need to be connected to the right person among all these GIGA experts, Karine Bebron, maybe Karine, you may switch on your camera. She's the coordinator of all the platform and she can guide you according to your needs. So never hesitate to contact Karine for any question that would be according to your needs. So, but let's focus today on the topic that we've planned, which is the preclinical imaging facilities related to this in vivo imaging platform at the GIGA Cyclotron Research Center. At the agenda today, we will first hear Mohamed Barry, who will give us an overview of these advanced technologies and cutting-edge equipment that are becoming more and more important in the whole process of preclinical and clinical studies, especially for the development of new drugs or novel tracers related to a very large range of application fields or pathology. And that's what we are going to hear. To present you a concrete application, we will welcome today three special guests. Frédéric Schins from ID Pharma, who will present a case related to intraocular surgery, and Michel from UCB Biopharma studying Parkinson's disease, and Ratish Raman from the University of Liège, studying the impact of osteoarthrosis on the bone density. So, to begin, it's my great pleasure to welcome Mohamed Barry, who is Principal Research Logistician at GIGA-CRC, Cyclotron Research Center Platforms. Mohamed is biomedical engineer and physicist. As Principal Research Logistician, Mohamed is sharing his time between the preclinical imaging platform and research, aiming to provide key insights to valuable in vivo test results for newly developed tracers and drugs, and also data processing for ongoing human studies within CRC research groups. And his main expertise is in PET neuroimaging, data acquisition and analysis, and kinetic modeling. So, Mohamed, please let us know about these CRC preclinical imaging facilities, giving advanced solution for a broad spectrum of application fields. And while you are preparing your screen, I suggest to our participant, if you have any question, please use the chat conversation tool. So, Mohamed, the word is yours. When you are ready, we are listening to you.
Thank you, Fabienne, for this nice presentation. So, hello, everyone. Thank you for attending this GIGA technological webinar. So, as previously mentioned, my name is Mohamed, and I will present you today our platforms, facilities, and the different services that the platform could offer. So, I will start this presentation with a brief introduction of the GIGA Cyclotron Research Center platforms, and then I will talk in details about preclinical platforms, the different equipment that we have, and the different services that we can offer. And I will, of course, provide some example of application in order to illustrate these two aspects. So then I will end this presentation with a small overview about the clinical platforms facilities. So to begin with, here you can see the GIGA-CRC platform organization. So we have 5 complementary sub-platforms working together in order to provide the process, the steps, the methods needed for drug development, disease, and treatment follow-up. So starting with research and development in radiosynthesis, this unit provide the process for design and production of radio tracer that is used in order to study a specific physiological, pathological, or biological process. Here I will open a small, let's say, a parenthesis in order to remind the definition of radio tracer. So radio tracer is a macromolecule labeled with radioactive isotope and designed to follow a specific physiological, pathological, biological process and provide the quantitative information that could help in order to understand this process. So here the right or the good example of that is the use of the fluorodeoxyglucose, the FDG, in order to monitor the normal and abnormal metabolism of the glucose in different field, and this radio tracer is used in different field and specifically in oncology and neurology. So here I close this parenthesis and I back to the organization of the platforms. So once the radio tracer designed and produced and validated or GMP validated by the radiosynthesis unit, the preclinical imaging could then offer let's say, wide range of imaging application that could follow that radio tracer inside the animal body non-invasively and could provide images and also quantitative data that could help to understand the disease, to follow the treatment and so on. So the validation of the radio tracer for human use is done on the human imaging platform. We have also, as you can see, the fourth sub-platform, which is a human electrophysiology platform or sub-platform, which has the needed knowledge and the facilities in order to do or record the electrophysiology signal in the human brain. So as we know that all these platforms generate a considerable amount of data that images and tables and all the kind of data. So all these data should be processed, analyzed, validated, and this is the task of the data processing and radiophysics unit, which take care of the computation modeling, mathematic modeling, kinetic modeling, statistical analysis, quality control of the data. So all these tasks are the task of the team of data processing and radiophysics. So these 5 sub-platforms are presented by preclinical and human or clinical platforms. So now I will talk in details about the preclinical imaging. So the aim, objective of the preclinical imaging is imaging and visualization of living animals for research purposes. It enables various real-time disease study, not only qualitatively by providing images, but also quantitatively. And this is thanks to the kinetic modeling, which provide quantitative data of physiological process. As an example, the receptor density, perfusion, and so on. So we work at molecular level, and this is thanks to the use of the highly sensitive molecular imaging facilities, which is the positron emission tomography. We can do longitudinal study, and this is thanks to the non-invasive aspect of the imaging solution that we propose. So disease progression and treatment response could be monitored non-invasively in the same animal, and this is of course reduce substantially the animal numbers used in the experiment. Preclinical or our preclinical imaging platform uses high resolution imaging modality, micro-CT, micro-MRI 9.4 Tesla, and highly sensitive technique like micropet in order to provide images and quantitative data of great value in key research areas such as infection, oncology, cardiology, and neurology. So now I will go through the different modality, providing some technical details about the different scanner that we have. So the first one is the micro MRI scanner Agilent 9.4 Tesla, dedicated to mice and rat, but also we can scan a bigger animal of the size of rabbit. So the system is equipped with receiving coils, volume, and surface coils that let's say able to cover the wide range of application. So the micro MRI system provide anatomical image, functional image, molecular imaging data, as you can see in these images. So the micro MRI could also provide quantitative measurement of a specific molecule in the tissue, and this is thanks to its spectroscopic capability. So the second imaging modality is the micropet scanner Focus 120 with its capability to record the data in list mode, which is of great help when you do dynamic study and you want to reframe your data. So the spatial resolution of this scanner, it's 1.5 millimeter, and it provides static as well as dynamic PET imaging of the brain, the heart, and any other mice, rat organs. So we can also do kinetic modeling for physiological and pharmacological parameter estimation like glucose consumption, receptor density, blood flow, and so on. So here you have the third modality, which is a 3D X-ray micro-CT scanner, which provide non-destructive, high-resolution 3D image of small laboratory animals, rat and mice, as well as a wide variety of biological samples, including mineralized and soft tissue density tumors, as well as archaeological and paleontological samples. So the resolution of this scanner is about 100 micron. So all these scanners are equipped with technical imaging unit for admission and extraction of anesthesia as we work in vivo in the animals. So we could also use this functionality of thermoregulation of the animals, and also monitoring of all vital signs. So our platform have also dedicated laboratories like HPLC, automatic gamma counter for tissue distribution measurement, as well as metabolite analysis in blood and tissue. We have also animal facilities, 48 ventilated rack for mice and 20 ventilated rack for rats. So after this quick presentation of the different facility that the platform have, I will go through some applications showing the use of our platforms or of our modality, imaging modality. So here you have some application extracted from a published paper. So the first one use the MRI diffusion technique and mathematical modeling to assess the surgery time after neoadjuvant radiotherapy. So briefly, neoadjuvant radiotherapy consists of providing some radiotherapy session before the tumor extraction, and this is in order to better control the tumor environment and to make the extraction successful. So the success of the tumor extraction is shown to be dependent on the time of the surgery after, of course, these series of radiotherapy session. So thanks to the diffusion-weighted imaging, MRI imaging, a correlation between the perfusion and the increased vascularization in the tumor was found. And then in order to extract successively the tumor without metastasis burden, the surgery then should be or must be performed out of the peak of the perfusion. So the image and that you see represent the MRI image of the diffusion-weighted imaging, as well as the computation done in MATLAB with our softwares here. So the second application uses the MRI technique to confirm the suitability of an optimized mouse model for vascularized lymph node transfer. So the MRI volumetric measurement done for these animals or for these models demonstrated let's say, new interesting data about the presence of skeletal muscle atrophy in relationship with lymphedema development. At that time, in 2019, the group was the first to report this new finding in mouse. So here in the image, you see the red color, which is the automatic extraction of the volume of the muscles. So then I have also two applications presenting or showing the use of the PET of the positron emission tomography modality. So the first part of the slide shows an in vivo PET-CT imaging model. So the aim of this study was the development of model for screening of novel PET tracer, a model low cost and quickly quick to set up. As you can see, it's eggs, chicken. In order to demonstrate the feasibility of this model, we tried the model of glioblastoma tumor in a chicken, and as you can see in the figures, there is this arrow showing the tumor in this model. So the second application. The main purpose of this second application is to understand how we can use the UCBH PET radio tracer in order to assess the synaptic vesicle 2A variation in vivo during the development of the tumor, the temporal lobe epilepsy in rat model. With this study, we demonstrated the feasibility of the suitability, for the the use of this UCBH PET radio tracer to detect differences between non-epileptic and epileptic animals. We were also able to follow up on the brain maturation and epileptic disease progression. So here I also will provide the two applications showing the use of the microcity. On this slide, the first study was published in Nature Communication. In this study, we aimed for growth hormone assessment. We used the microcity scanner to assess the body volume composition, and as you can see in these figures, the red color shows the automatic extraction of the fat in the mouse, and you can see also the modification in the bone showed by CT. So the second study in this slide demonstrates the use of the microcity in the non-medical domain. In this archaeological study, we used microcity to assess the turbidity in clay with the proposed to study the evolution of sediment. So figure A and B demonstrate the ability of the microcity to differentiate between the sediments of two different sites. So to summarize, through the presented application and the references provided on this slide, I hope I was able to clearly stress the utility of our tools and their potential, as well as our capability of answering novel research questions. So you can also find detailed information on the GIGA platform website. So now regarding our clinical platform, I will talk about the facilities that we have. We have two MRI scanners. The first one is a 7 Tesla whole body imaging, which is the first 7 Tesla MRI scanner dedicated to research in Belgium. So this MRI 7 Tesla provide anatomical high resolution imaging with a sub-millimeter resolution, and we could also have a functional imaging with this modality. We have also a second MRI scanner, 3 Tesla whole body imaging. The modality could be or is used for anatomical imaging and functional imaging. So we have also the third imaging modality, which is a PET camera, HR plus Siemens, high resolution 4.6 millimeter, dedicated only to research, and it's also whole body PET. This camera could, of course, provide static and dynamic image. These imaging modalities are used to assess brain correlates for healthy and pathological aging. So, taking it all together, we have the platforms and nodes enabling us to provide imaging services solutions in preclinical and clinical area. Regarding now the radio tracer development, the whole procedure can be done in our center, starting with the radiosynthesis and ending with the clinical trial. So thank you for your attention, and I would like also to thank all my colleagues. I would like also to thank people who will share their experience as user of this platform, and they will talk. Their talk will follow mine. Thank you very much.
Thank you very much to you, Mohamed, for this very complete presentation of the platforms. And for a first concrete case using these facilities, especially the MRI, I'm pleased to introduce you Frédéric Schins, who is a non-clinical development expert for the company ID Pharma. As background, Frédéric is doctor in vet medicine. He has a PhD in virology and vaccinology, as well as a master in laboratory animal sciences from the University of Liège. From 2002 to 2007, he was director of the virology department and scientific coordinator of the CER group, which is Applied Research Center dedicated to health and agriculture in the city of Marche, south of Belgium. And from 2007 to 2013, he handled different positions at GSK Vaccines in Belgium, such as the R&D leader, project leader, and director for development and competitive intelligence. He's now freelance since 2013, acting as a consultant for private companies and non-governmental organizations, assisting them in the non-clinical development of vaccines, cell and gene therapy, medicinal products, classical medicinal products, and medical device. He's here representing the company ID Pharma with a case of intraocular implant visualization by MRI. So, Frédéric, I invite you to share your screen and please tell us more about this application.
Thank you very much, Fabienne, for this nice introduction. I will put it in slideshow. Open it. Okay, let's go. As a first step of my presentation, I will say some words about ID Pharma and the reason why ID Pharma initiated a collaboration with the CRC platform. As a second step, I will present some MRI results we have generated on rabbit eyeballs implanted ex vivo with an intraocular implant. Then ID Pharma has been created in 2012. Its headquarters is located in Liège, and the company is quite fast growing, let's say like this, since now around 80 people are employed. ID Pharma is acting from the R&D department to the GMP manufacturing with UniD manufacturing, which is specialized in sustained drug release in micro dimensions. In addition, ID Pharma is also a distributor of medical devices used by ophthalmology surgeons. All the activities of the company will be hosted in a specifically dedicated building, which is under construction in the Liège Scientific Park. And the integration of all these activities in this new building is planned for 2022. The usual administration route for the majority of ophthalmological treatment remain via eye drop instillation. Aiming at improving safety and efficacy of that usual administration mode, ID Pharma portfolio comprises 3 products: Timody, Combody, and Inflamady. Timody is a non-biodegradable intraocular implant delivering timolol to treat glaucoma. Combody is a non-biodegradable implant delivering controlled doses of 2 active pharmaceutical ingredients. And finally, Inflamady is a biodegradable short-term implant based on a well-known drug designed to prevent inflammation after eye surgery. I will now detail more deeply Timody on the next slide. Timody aims to reduce intraocular pressure to to treat patients with glaucoma or ocular hypertension. In contrast to twice daily administration of timolol eye drops, Timo-D will be implanted intraocularly and deliver timolol for a period of 2 years, with the possibility to replace it by a new one. Timo-D will then answer two important unmet medical needs for patients treated with timolol eye drops. The first unmet medical need is to increase the safety of timolol treatment, since after timolol implantation, which is in contrast to timolol eye drop, timolol systemic effects will no longer be observed, since timolol is not present in the blood after Timo-D implantation. In addition, Timo-D will increase patient compliance, especially in elderly people, for whom it's sometimes really difficult to perform a self-administration of eye drops twice per day. Timo-D will enter into clinic soon, early 2022, early next year. So we started a collaboration with the CRC platform to investigate the feasibility to use MRI to visualize Timo-D after surgical implantation. Indeed, it's important for the ophthalmologist to have methods enabling him to visualize, localize Timo-D during all the implantation period and just before explantation to improve the surgical explantation. To assess MRI ability to achieve this, we developed a model on rabbit eyeballs to test different MRI parameters. Rabbits were chosen since exhibiting an eye size similar to the human eye size, accommodating the Timo-D dimensions. Since the design of Timo-D, and it's unfortunate for you today, since that design is confidential, I will then show you the result generated with the CRC platform with a normal rabbit eyeball and a rabbit eyeball implanted with an artificial intraocular lens used to treat cataract. But what is a cataract surgery? As you can see on this picture. That surgery consists in the replacement here of an opacified natural lens by an artificial lens, as you can see here. To achieve this, a corneal incision is performed, allowing surgical instrument to reach the lens. The core of the lens is broken into small pieces and then removed. Only the capsular bag, the external part of the lens, will remain, in which an artificial lens is implanted, restoring the vision acuity of the patient. At CRC, we first performed MRI on a normal, let's say, eyeball, rabbit eyeball, as depicted in this picture. Selection of some parameters allowed us to visualize the desired eye compartments, as you can see here, and in different axes, and also in 3 dimensions. You can see here the cornea, the anterior compartment, the iris, the lens, and the posterior cavity of the eye with the vitreous, and here the optical nerve. And the power of the technique is that based on the acquisition, you can then generate after the acquisition all the plane you want to generate. And you can also, let's say, have a trip inside the eyeball within specifically dedicated IT instruments. Then let's look at some views we generated in the frontal plane, meaning from the front of the eye to the back of the eye. And you can see here a rabbit eyeball with its natural lens from the front of the eye to the back of the eye. And you can see here the lens, the natural lens, this gray circle. And on the and below, you can see the same types of picture showing the intraocular lens that is implanted in the capsular bag during the cataract surgery. And you can, let's say, have that trip also inside the eye from the front of the eye to the back of the eye. And you can clearly show here, see here that we were able to visualize that intraocular implant. I have not shown you that today for confidentiality reasons, but the feasibility to see the details of our TMOD intraocular implants was also confirmed in a set of additional experiments. Then, as a conclusion, and thanks to the CRC collaboration, ID Pharma was able to demonstrate that MRI is a useful imagery method to visualize ex vivo ophthalmic implants implanted in an eyeball. Knowing that, the next steps, we will now assess the feasibility to use MRI to visualize TMOD implant in living animals with the aim to translate the technique to humans implanted with TMOD. In addition, we will continue to use MRI as a very useful and interesting imagery technique in future ID non-clinical development plans. And that's the end of my presentation. Thanks for your attention.
Thank you, Frédéric, for this, for sharing your experience and a very interesting concrete case. So to present the second case study focused at the time on the PET scan equipment, we will have the pleasure now to welcome Anne Michel from UCB Biopharma. As background, Anne has a master followed by a PhD in psychological sciences from the University of Liège, training as neuropsychologist. Her area of expertise are neuroscience, preclinical drug development, and neurodegenerative disorders. She joined UCB Biopharma 20 years ago, and she's currently director in vivo lead, leading a team of scientists in charge of the preclinical activities to support the in vivo projects in neurodegeneration. One of the major activities is the development of rodent models and their characterization to ensure the adequate testing of potential new therapeutics. So Anne, thank you very much to having joined us today. The word is yours. We are listening to you.
Thanks, thanks, Fabian, for the introduction, and hi, everyone. I'm going to talk to you indeed about the CRC-UCB collaboration, scientific collaboration that we had together for the development and optimization of two preclinical models of Parkinson's disease. As an in vivo lead in pharma industry, one of my major objectives is to ensure the right development of the preclinical rodent models. To do so, we are facing many issues and challenges. Identify the preclinical in vivo models relevant for the pathophysiology of the disease of interest is key to ensure optimal testing of our therapeutics. What is at stake in my field is the selection of meaningful readouts and biomarker that will improve the translatability and predictability of the in vivo models, and then with the ultimate hope of reducing the attrition rate. To do so, UCB and CRC set up a scientific collaboration where UCB brought its expertise in preclinical models of in vivo, and CRC brought its experience in rodent in vivo imaging. More concretely, UCB oversaw the behavioral assessment, the tissue collection, and the subsequent biochemistry and immunohistochemistry analysis, whereas CRC took in charge the microPET study to assess 2 different tracers in the UCB rats. Before to go ahead about the detailed explanation of our collaboration, I really would like to set the scene about the pathophysiology of Parkinson's disease for those who are not familiar with it. As you can see on this picture, Parkinson's disease is mainly associated with 2 major characteristics. First, with the loss of pigmented dopaminergic neurons that you can see here in the substantia nigra, and second, with the presence of intraneuronal inclusion called Lewy bodies, and that are mainly composed of one specific protein, which is progressively becoming pathological, the very well-known alpha-synuclein. According to the Brack hypothesis, alpha-synuclein is becoming progressively pathological via spreading and seeding mechanisms. The dual hit hypothesis states that sporadic PD start in 2 places, in the neurons of the nasal cavity and in the neurons of the gut, and then the pathology would join the CNS via the vagus nerve, would reach lower brainstem region and join the substantia nigra, and eventually would invade the whole cortex at more advanced stages. To reproduce this feature of Parkinson's disease pathology with rodent, we have access to several rodent models. In particular, 2 models are based on the administration of toxic substance directly into the brain of the mouse of the rat to really recreate the pathology. On one end, the toxin model is recreating the profound dopaminergic loss into the substantia nigra. The dopaminergic lesion is created via the direct brain ejection of one toxin, which is called the 6-hydroxydopamine, and which is producing an immediate destruction of the dopaminergic neurons, leading to a severe and permanent dopaminergic loss and to a strong motor phenotype. This model, this rodent model, is a very acute model. Besides this immediate model, there are no more recent approaches that are more progressive in the sense that they are recreating the very progressive alpha-synuclein spread and the progressive dopaminergic degeneration, mimicking really more the human situation. Amongst those progressive models, the AAV-induced A53T alpha-synuclein expression is a model of choice to really approach this question. In the work done in collaboration with the CRC, we have decided to join our effort to characterize these 2 models, and we collaborated on 2 main studies. A first study had for objective to compare 2 dopaminergic PET tracers, the FMT and the fluorodopa, in the toxin rat model bearing partial or severe dopaminergic lesion. As a result, we showed that FMT was more sensitive than fluorodopa to investigate the dopaminergic terminal loss. In a second study, we decided to use the FMT PET in a progressive model of Parkinson's disease, the AAV-induced alpha-synuclein model. The experimental design is on the next slide. In this very large study, we had 2 main experiments. One aiming at characterizing really the model for behavior, biochemistry, and immunohistochemistry, and it was done at UCB. The second experiment was done by CRC, and this had for objective to test AAV A53T alpha-synuclein rat in FMT PET. This was really a real close collaboration between the two teams for these two longitudinal studies. The results were very interesting in the sense that they show that in this model, the A53T, the longitudinal monitoring of the progressive functional dopaminergic deficit was possible by combining both FMT imaging and motor behavior. As you can see on the left panel here, the picture really clearly shows the progressive decline of the FMT signal over time. So here it was recorded at 3, 5, and 12 weeks. And we can really see this progressive disappearance. In parallel, when we monitor the progressive deficit at different time points, you can see here that there is a progressive reduction of motor capabilities for the rat. This second study was published in Neurobiology of Aging. So to conclude, both UCB and CRC joined their expertise to characterize, develop relevant preclinical model of Parkinson's disease. The collaboration between the two teams was excellent, and the interaction really led to 2 scientific publications. If you have any questions, don't hesitate to reach me out. And thanks a lot for your participation.
Thank you very much to you, Anne, for sharing this experience with us. The third and last case of our program today is a study on the PET scan, aiming to better understand the impact of osteoarthritis disease on the bone density. And therefore, I have the pleasure to welcome Ratish Raman, who is early stage researcher pursuing currently a PhD at the Laboratory of Organic and Regeneration at the University of Liège. Ratish is coming from India, where he completed the Master of Science in Biochemistry at colleges affiliated to Mumbai University. He also has graduated from Uppsala University in Sweden with a Master of Medical Science in Molecular Medicine. In his PhD, he employs zebrafish as a model system to study skeletal development. So please, Ratish, tell us more about your project using micro-CT to study fish skeletal biology. Put on your microphone so that we can hear you. And you can...
Yes, just a second. I'm sharing my screen.
Please.
Oops, sorry. Stop presenting. 1 minute. There was a confusion. Can you see my screen?
Yes. You just put the slideshow and it will be okay. We're listening to you, Ratish.
Perfect. Thank you so much for this opportunity. I would be giving a case example of using micro-CT to study fish skeletal biology. I am a PhD student here at Liège. So this is a maximum intensity generated image of an adult zebrafish skeleton. And as a part of my PhD, I'm trying to study the role of certain genes whose proteins have been identified in human osteoarthritic patients. So the whole project is to study the role of those genes in skeletal development. And so let me go to the next slide. So I would like to start by talking about the importance of the zebrafish as a model system, because there are several model systems that are employed to study in biomedical research, and zebrafish is a small model system that has got many advantages, being that it's been widely used as a system to study the development, the regeneration, and especially in the disease of skeletal in terms of the basic and preclinical aspect. Given the fact that it has a 70% genomic similarity to humans, it becomes even more important to employ this model system because of its small size, being very good in rapid development. It is an inexpensive way to study. You can have a lot of eggs that you want to study the early developmental stage. You can easily maintain them. You can perform forward genetics and reverse genetics by, for example, in reverse genetics, you can use genome editing like CRISPR-Cas9 to generate mutations on the target gene of interest and then study the role of those genes on your topic of interest. And for example, here, as a matter of fact, that we are using is to study the role of genes in skeletal development. So as an early stage, here I present to you a sample image as a comparison that you can do cartilage stainings in blue compared to the mineralized bone or tissue in red, which is alizarin red, to study the bone and the cartilage elements. And you can see that you have on top the embryonic development stages of the mice at various stage compared to the zebrafish. And when you see from the early to the late, the elements are morphogenetically different. And hence, if you're looking at the adult fish, it becomes very important to look at the skeleton. Now, the zebrafish skeleton, as shown in this paper, clearly gives you a picture that actually shows you the skull element, the abdominal, the caudal, and the tail fin elements. And what makes it more important is that you have around 22 bones in the like you have 74 bones in zebrafish compared to 22 in humans or in mammals in the skull element area. And in the spinal side, when we go, that is over here, you have around 30 to 32 vertebrae in the zebrafish compared to 33 in humans. And you can study these various elements. For example, the vertebra, as depicted here, you can look at the thickness, the vertebral length as shown here. So I now start with showing you the examples that you can do, which is like you can perform X-ray scans using Cyclotron, which clearly shows between your control and the mutants, just to have an explorative way, because then you can identify some skeletal malformations as in the figure shown and the mouse here hovering on the left side, wherein you have a malformation here towards the caudal part. So then you can, when you perform the micro CT, you can actually go on the specific part by cutting it out, or you can perform the micro CT on the entire organism as well. Like, for example, this is a zoomed in view of the malformation from the X-ray scan that was obtained. And then I present to you an example of a micro CT scan and then performing a 3D reconstruction and generating a video. And this was done with the help of the CRC bioimaging platform with Mohammed here. And that's what we approached them to get their assistance and help. So I'm very thankful for that. And I present to you an example that you can perform various analysis. As I mentioned, you could see them on the right side. You can have three different sectional views: the coronal, the sagittal, and the temporal. And then in any of the sections, you can go for looking at the thickness and the length. And here is on the left side, on the right side, you can see the 3D maximum intensity projected image of the sliced vertebra that you focus on. And then you can perform the analysis compared to the control and the mutant, because when you are going to have a study, like in case of my project, you are looking at the aspect of a control and a mutated zebrafish, because we are trying to study the role, as I said earlier. So it is always important to have your control fish and a mutated fish to get a better understanding of the skeletal morphology and looking at the element up close, which is possible using X-rays or micro CT. So this is one example showing the measurement of the thickness and here of the length. And here, this is one example that For example, there is a gene in my project that was identified in an osteoarthritic patient. And while I was trying to do the vertebral analysis, I observed certain important observations, like you can see here from the osteoarthritic spine and a healthy spine here. The difference is that an osteoarthritic spine has burn sparings, which reduces the intervertebral space. And you can clearly see in this 3D projected image that we see a similar kind of observation. And you can compare it with the wild type to be sure that these are not just events, but these are really specific events related to a condition. And the major importance that brings me to highlight is that the reason we are also collaborating is that we are also interested to look at the tissue mineral density or the bone mineral density. And in order to do that, we are collaborating with the CRC here, wherein using a MATLAB approach, there is a software called the Fish Cut that that uses this MATLAB to study and calculate the bone volume and the bone mineral density. And it is for this reason I'm here at the CRC with Mohammed, Christian, and Professor Alain Pellenwood. I must thank them for a valuable collaboration, which brings me to the end to give acknowledgements to a lot of people that are mentioned here, especially to our collaborators in Ancona, and also in Paris, in INSERM, for their valuable help in doing micro CT scans and Faxitron scans, which I did and got back the images. And with the CRC platform, we are performing the analysis. So thank you so much to my lab members and my professor, Mark Muller, for valuable help, and my co-supervisor, Professor Eve. Thank you.
Thank you very much to you, Ratish, for this witness. So it's time to close our public session. To respect the agenda, I address a special thanks to our keynote speakers for their availability and the really relevant and concrete information they have shared with us today. So again, thank you. Thank you also to my colleagues in the back office, having taken part to this organization. So Aurélie, Karine, Annick and Florence, and thank you to you, of course, for having joined us today. I'll remind you that Mohamed Barry and Karine Bebron are at your disposal, so don't hesitate to contact them to ask for a meeting or to talk about your specific matter. This session has been recorded, and Aurélie will, of course, send you a link to review or to share it. So thank you again. I address you my best wishes for the coming year and success, of course, for your own projects. We'll be back with a webinar on the platform Immunohistology on January the 27th, and in between, best wishes for you.